Thank you very much, Nene. Let me try again hopefully this time it works like it did. So let's see. Can you see?
Yes.
All right. Can you see my screen now?
Yep.
Perfect. Thank you, Nene, for the introduction. I'm Oumie. I'm doing my PhD at the Institute of Evolutionary Biology at the University of Edinburgh. So today we'll be talking about discovering the fascinating world of viruses. I just thought that before I start, I just take you through my CV. So I did my A-levels in the Gambia at Marina International School, and I finished in 2013. I did it in mathematics, biology, and chemistry. So from 2014 to 2017, I did an undergraduate degree in biochemistry at University College London, which was jointly funded by the MRC unit, the Gambia, and the Medical Research Foundation. As you can see, there's a 1-year gap here between when I finished my A-levels to starting my undergrad. What happened was that after my A-levels, I got an offer to study biochemistry at UCL, but like a lot of Gambians, I could not afford to do so, and University of the Gambia was not offering biochemistry at the time. So I took a year's gap during which I worked in a bank, and it just happened that the MRC was looking for a Gambian to study any related course at any UK university, so that worked out nicely for me. In my final year undergrad, I was offered a Wellcome Trust Host Pathogens and Global Health PhD program by the University of Edinburgh. And as part of this PhD, I had an MRes in Global Health as part of the first year, and I'm currently doing my PhD in virus evolution. Last year, I was awarded a Verley Gradwell Traveling Fellowship in Insect Ecology by the University of Oxford, and I'm currently doing that as part of my PhD. So today I'd like to divide the talk into two parts. The first part will talk about viruses in general, and the second part, I'd like to delve more into my PhD and how that fits into viruses. We're currently in a global pandemic. Everyone's been talking about coronavirus, panic buying, lockdowns, wearing a mask, and all this chaos was actually caused by a virus. So firstly, let's discuss, what are viruses? I will describe viruses as the smallest, most abundant, and most diverse infectious agents. There are currently 10 non-million viruses in the world, which is 10 to the power 31. That number does not make sense in my head, but if we want to think about it, there are actually more viruses in the world than there are stars in the universe. Viruses are very small, actually. They have a diameter of 20 to 250 nanometers. There was an article that came out last week that said that if we actually put all the coronaviruses in the world can fit in a can of Coke. That's amazing. Viruses can infect all forms of life, but they can only replicate and survive inside a host cell. The inability of viruses to survive without living organisms is a fascinating topic for scientists. So now that we know what viruses are, one of the most interesting discussions in evolutionary biology is, where do viruses actually come from? And this discussion is stemmed from the discussion, are viruses living organisms or not? Some scientists say yes, and some say no. And this is because they cannot survive outside the host. I personally am of the school of thought that viruses are not living organisms, but rather they are biological entities that can cause disease. Another thing is that virus evolutionary history is inferred from today's viruses, as they do not fossilize. So to answer these questions of where viruses come from, evolutionary biologists have come up with 3 running virus origin hypotheses. The first hypothesis, called the progressive hypothesis, which is that viruses actually arose from mobile genetic elements. The second one, the regressive hypothesis, is that viruses are actually remnants of cellular organisms. The third one, called the virus first hypothesis, is that viruses actually prevade or co-evolve with current cellular hosts. So now that we know what viruses are and have an idea of where they come from, let's look at the structure. Viruses contain a single nucleic acid, which can be RNA or DNA. RNA is like DNA, another way that cells store genetic information. This nucleic acid genome can be single-stranded RNA, double-stranded RNA, single-stranded DNA, or a double-stranded DNA. Some virus genomes are segmented and some are not segmented, just generally some exist as chromosomes, we can call it that. This genome is surrounded by a protein capsid that protects it from the environment. In addition, some viruses like the coronavirus actually have a phospholipid envelope. Others, like the poliovirus that causes polio, does not have this phospholipid envelope. Viruses can also come in a variety of shapes. Some have head and tail, some are filamentous, etc. So now that we've looked at the structure, let's look at the life cycle of a virus when it gets into the cell. This can be divided into 3 steps. The first one, entry. The virus needs to enter the cell. This occurs by the virus surface proteins. The virus receptor surface proteins must recognize and bind the host receptors. Like here for coronavirus, the S protein needs to recognize and bind the host ACE2 protein. And when this happens, this binding triggers viral uptake and fusion. And now that the virus has entered the cell, it needs to go to the second stage called replication. The virus uses the host machinery to actually replicate its genome. However, the mechanism and extent to which this host machinery is relied on varies between virus families. Now that the virus has replicated its genome, it needs to exit the cell. So the assembled virus genomes are then released from the cell, and this release could be via cell lysis or exocytosis. So we've looked at viruses more generally. Now let's delve into emerging infectious diseases. Everyone's been talking about it since the whole COVID. So many emerging infectious diseases are actually zoonotic. Zoonosis means is any infectious disease caused by a pathogen that has jumped from an animal, which is usually a vertebrate, to a human. This is known as host switching because the virus switched host from using this vertebrate animal as a host to now having the host as humans. This host switching may occur through direct contact with the animal, contaminated food, or water, or through vectors. Current research has shown that climate change can be a contributing factor. And 3 important viral diseases that I think that are as a result of zoonotic host switching is HIV, measles, and COVID-19. These are quite important because HIV is one of them, I'll say, the most important viral disease in the world. Measles and COVID-19 are all quite important too. We'll see why in the next slides. So let's start with HIV. HIVs are lentiviruses. Lentiviruses are RNA viruses that cause chronic infections in mammals that is followed by a long period of latency. HIV infection occurs via bodily fluids and is the main cause of AIDS. Currently, over 38 million people are living with HIV and AIDS. Old world monkeys are naturally infected with over 40 different lentiviruses, which are known as SIVs, simian immunodeficiency viruses. So this is a paper that we're going to look at. It was published by Paul Sharp, who is the head of my institute in Edinburgh. So HIV has 2 lineages, HIV 1 and 2. And Paul Sharp was able to show that this was due to 2 independent zoonotic transfers of SIV infecting African primates. HIV 2 is restricted to West Africa and it could be of Sudi-Mangabe origin. HIV 2 is mostly in Guinea-Bissau and Senegal, but it's currently being replaced by HIV 1. So now let's look at HIV 1 more in depth. HIV 1 has M, N, O, and P groups, and all of this was as a result of independent cross-species transmission. HIV 1M is the principal cause of the AIDS pandemic. M and N are of chimpanzee origin, that is, they jumped from chimpanzees to humans, and this may have occurred in present-day DRC. Kinshasa may actually be the cradle of the HIV-1 pandemic. It is believed to have circulated in Kinshasa for 50 years before its identification in 1983. This is because there were some blood samples that were collected in the 1950s in Kinshasa, and when they were sequenced, they actually found that by that time, HIV-1 had already diversified into its current present-day lineages. HIV-1P is of gorilla origin, but the region is unknown. Group O could be of gorilla or chimpanzee. All this information I've talked about can be found on here, the phylogenetic tree. Let's spend a minute looking at how we interpret phylogenetic trees, because there'll be a couple more coming. So this is called a phylogeny or phylogenetic tree, and literally, basically, it just shows the relationship between organisms or their evolutionary relationship. So if we look at this here, this is a clade, and these are the tips of the tree. If we go back horizontally, we find a node, and the nodes actually just show the ancestor of these two. The vertical distance between tips does not matter, but we look at the horizontal distance. If it's a time scale phylogeny, it can show us the evolutionary distance since the ancestor. So here, this is HIV1M, this is N, this is P, and this is O. As we can see, these are more clusters with this chimpanzee, this one clusters with gorilla, and O could be of gorilla or chimpanzee origin. So now that we've looked at phylogenetic trees, let's move on to the next zoonosis disease, measles. Measles is a contagious disease caused by the measles virus. It is transmitted via respiratory secretions. Measles causes rashes, fever, and can lead to blindness and death. A precursor of measles began causing infections in humans as early as the 4th century BC. It is believed that measles may have evolved from rinderpest that infects cattle. Rinderpest has been extinct due to vaccination. However, in humans, measles infections have been decreased by 74% due to vaccination. But still, even though it is a preventable disease and the vaccine works, over 100,000 people die each year due to measles. And there's even measles resurgence due to anti-vaccine sentiments. If there's one message that we should take away from this slide, is that vaccines work. And if you're offered a vaccine, take it and do not fall trap into the anti-vaxxers campaign. The third one we'll be looking at zoonosis is COVID-19. Coronaviruses are a diverse group of viruses infecting many different animals. SARS and MERS emerged in 2002 and 2010, respectively. COVID-19 is caused by SARS-CoV-2. SARS-CoV-2 is a highly transmissible and pathogenic virus. This is a paper co-authored by Andrew Rambo, who's also in our department, and they've been able to show that the SARS-CoV-2 in human actually jumped from bats to human. This is because there was 96% sequence identity to a bat cervicovirus, rat G13, which was sampled in 2019. And this is the bat virus, and this is the human, and it's believed that it jumped from bats into humans. So we've looked at host switching and using 3 important viral diseases as examples: HIV, measles, and COVID-19. An important host switching does impact an important feature of viruses called virulence. As I've mentioned, many emerging infectious diseases are due to host switching, and virulence is the amount of damage a pathogen does to its host. Virulence can actually increase or go unnoticed following host switching. Evolutionary theory states that virulence couples with viral load, creating an evolutionary trade-off. As a result, high virulence increases transmission per unit time, but will kill the host faster. If you think about it, it makes sense. Since virulence is the viral load, that is the amount of virus there, if there's a lot of the virus, it replicates a lot, and you have a lot of it, it will be transmitted quicker per unit time. However, if there's too much, if it happens too quickly, the host will die. So when intermediate levels of virulence are optimal for pathogen fitness, and this is when the virus can replicate in a way that's fast enough to make sure that it's transmitted, but not too much that it kills off its host. So since host switching impacts virulence, we need to ask important host switching questions. Number 1, what is the host range of a virus? That is, how many different species can a virus infect? Number 2, we need to answer, why are some host switching dangerous and others go unnoticed? We're constantly being exposed to our environment and to vertebrates. And obviously, we're getting viruses from them, but some go unnoticed and some cause a pandemic like the COVID-19 we have currently. Thirdly, why are some viruses undetectable in their native hosts, but very virulent when they jump to new hosts? Like the example I gave, the Zika virus was in bats as early as 2013, nothing happened, but when they jumped to humans, they caused the present chaos. Number 4, does host-relatedness influence the success of host switching? What this is asking is the evolutionary relatedness between hosts. So our closest relatives, chimpanzees, HIV jumped from chimpanzees into us. So does that influence the success of host switching as compared to, let's say, a cabbage, which were evolutionarily more distant to us compared to chimpanzees? So to answer this question, we need to do it in the laboratory. So let's just remember 3 points. For a host switching to be considered successful, the new host must first be exposed to the virus. Secondly, the virus must be able to replicate in the host. And thirdly, there should be sufficient onward transmission of the virus. But to study this in the lab, to answer the questions that we mentioned earlier, we cannot use humans to study viruses in the lab because that is highly unethical. Fortunately, there is a family of insects called the Drosophilidae. So the Drosophilidae family can be used to study host switching in the lab. This is because they are cheap and very easy to maintain in the laboratory. Their innate immunity is well understood, and we can use them to study the relationship between host switching and phylogeny. So a PhD student in our lab at the Ben London, who was a PhD student in our lab, but he's now a PI at University of Exeter, he worked on this whilst he was in our lab. So he worked on Drosophila C virus, which is an RNA virus that naturally infects Drosophila melanogaster, which I highlighted here in the phylogenetic tree. So he took 48 species of the Drosophilidae, which share a common ancestor about 20 to 50 million years ago. So he infected the flies with DCV, and he measured virulence. He did this by measuring the mortality following infection. He measured viral load, which is the quantity of the virus following infection. And then he measured transmission by exposing uninfected flies to infected flies. So what did Ben London find? He found that distance from host species does not determine virulence. If we look at this tree here, low virulence is green and goes all the way to red, which is high virulence. For example, Drosophila melanogaster is the host species for the similants here, and these ones where Drosophila virulence is evolutionary closer to D. melanogaster as compared to, let's say, Drosophila nebulosa. But the virulence that's found in Drosophila nebulosa is closer to Drosophila melanogaster than Virulence here. So hence, distance from host species does not determine virulence. But he found that closely related species have similar virulence. Like if we look at here, these ones cluster, have high virulence, and these clustering here have a lower virulence, which makes sense because the virulence differences could actually be due to differences in the virus ability to bind host receptors and host antiviral response. Closely related species will obviously have more similar host receptors and more similar antiviral response. Hence, they'll have similar virulence. And he found that virulence was tightly coupled to viral load, and he also linked high virulence to higher transmission rates, which is consistent with the evolutionary theory of virulence being coupled with viral load. So now that we've looked at Drosophila and virulence and host switching and also viruses, I'd like to move on to the part 2 of my talk, which is my PhD that I'm doing at the Institute of Evolutionary Biology. So this building is where I work. My office cannot be seen here, but it's down at the back there. And let's go. So my PhD is mostly focused on discovering viruses because we need to discover the viruses to be able to isolate them and do work in the lab. So when we think of a scientist, we picture a wet lab scientist like this lady here with a lab coat and mixing chemicals and having all those fun colors. There are the scientists that are called bioinformaticians who use computer programming to answer biological questions. So my PhD, I'm about 80% into bioinformatics and 20% of the time I spend it into the lab. So this is what a typical day looks like for me, just working on the computer. Who am I kidding? This is more accurate. A lot of the time I find myself wanting to smash that computer because my code is failing. However, when I get it to work, it's always very satisfying. So let's look at my PhD more, virus discovery into Drosophilidae. So how do we discover viruses? We can use metagenomic sequencing of wild Drosophilidae for virus discovery. Metagenomics is the study of genetic material recovered directly from environmental samples. To date, over the last 15 years now, over 120 Drosophila viruses spanning over 25 families have been discovered mostly by our lab. So Drosophila is a species, it's a genus under the Drosophilidae family. So how do I go about discovering these viruses? I collect flies from the wild, I extract their RNA, I do RNA sequencing on them, and then I look for virus sequences. So what are the questions that I'm trying to answer? Number one, I want to know what viruses infect African Drosophilidae. Number two, once I find these viruses, I want to know how diverse and prevalent are these viruses and what Drosophilidae species do they infect. Thirdly, I want to know how do different sequencing technologies compare in metagenomics virus discovery. I want to compare direct RNA sequencing by Oxford Nanopore Technologies to Illumina sequencing in metagenomics virus discovery. Another question that I want to know is how does virus infection change the gene expression of the Drosophilidae? And for this, I am using the most studied member, Drosophila melanogaster, as a model organism. So why am I focused on African Drosophilidae? Well, the most studied member, Drosophila melanogaster, has its roots in the African continent. It's believed that Drosophila melanogaster moved out of Africa over 10,000 years ago. This was around the same time that 10,000 years ago, the Drosophila melanogaster became a human commensal. This commensalism may have occurred in present-day Zimbabwe in the Matapos rainforest. The hunter-gatherers that occupied the region ate the same fruit, the marula fruit, as the Drosophila melanogaster at the time. And also research from our lab. So this paper was published by our lab in 2015, and it shows that African Drosophila viruses have a deeper evolutionary history than their European, Northern American, and Australian counterparts. So this is an evolutionary tree here. This is a phylogenetic tree here. And the ones that I highlighted in green, this is all sequences of La Jolla virus. The red ones are ones found in the species called Drosophila similans. The blue are the ones found in Drosophila melanogaster. So these ones that are highlighted in green are viruses found in Drosophila melanogaster collected from Accra in Ghana. All the other ones are the ones collected from America or Europe. As we can see, the ones from Accra have a deeper evolutionary history. Hence, we can conclude that African Drosophila viruses will actually reveal novel virus sequences, associated prevalences, and host range. So what do I do on a typical collecting flies? This is me in a village called Jambur in Gambia collecting flies. So once I collect the flies, I take them to the lab to sort them, sex them, because to get the males out and then sort them into species and sexes. And most importantly, when I go on field work, I eat lots of mangoes. I actually think I did more of the third one than the first two, but don't tell my supervisor. So I did mention a lot of questions that I'm interested in for my PhD, but one of them, all the others I'm like working on them, but they're not complete. But this chapter, we have a more concrete answer, which is how do viral infection change the gene expression of Drosophila melanogaster? Well, viruses change Drosophila melanogaster gene expression in a myriad of ways. For example, when female flies are infected with some viruses, they actually produce less eggs, which makes sense if we think of evolutionary life history trade-off. The flies are actually investing more energy into survival, which is getting rid of the virus, than reproduction. However, most studies on gene expression in response to viruses inject non-native viruses into Drosophila melanogaster. The infection route and non-native viruses can affect the validity of results. If we think about it, the flies are not being injected in viruses in the wild, and they're not encountering non-native viruses like mosquito viruses, like Synvis virus. So by using non-native viruses and injecting them can actually lead to inconclusive results that do not represent what is found in nature. Fortunately for me, there are many publicly available RNA sequencing data that other labs around the world have done on Drosophila melanogaster and put in the public database, but they did it for other purposes. So I can use this data, reroute it, and answer the questions that I'm interested in. So what questions do I want to ask about gene expression in Drosophila melanogaster? Number one, I want to know what viruses are naturally present in laboratory Drosophila melanogaster. Two, how does the gene expression change in response to infection? 3, is the gene expression between males and female flies actually different? 4, does the age of the fly affect its gene expression? 5, do virgins and mated female flies respond differently to virus infection? So this is the bioinformatics pipeline I'm using to answer those questions. So I get the RNA sequencing from public database. I separate the virus and fly sequences. So I have virus sequences here and fly sequences there. Then from these sequences, I then determine the flies that are infected with viruses. Once I determine the flies that are infected and those that are not infected with viruses, I then compare the gene expression of each gene before and after infection. So what have I found so far? Well, I found that Drosophila melanogaster viruses such as Galbert, Mottsmill, Zika, and Nora are found naturally in laboratory D. melanogaster. Interestingly, some of these viruses such as Mottsmill and Zika have not yet been isolated, which is great because it means that we can actually know what is happening, how the flies respond to these viruses that we don't even have isolates for. I also found that males and females respond differently to virus infection. And I found some changes in immune response genes that sometimes when flies are infected, some antiviral genes like the RNA interference genes go up, which makes sense because the flies are responding to the viruses. I found no effect of age and mating state on how flies respond to viruses. And I'm currently tidying up the analysis and writing a paper. So on that note, I hope I've been able to convince you that viruses are the most fascinating biological entity in the world. So I'd like to thank you, all of you here, for listening, of course. Our lab, my supervisor, Darren, our 2 postdocs, Fergal and Yuguan, Megan, another PhD student, and Paul, our research technician. I'd like to thank the Health Science for the Gambia and the University of the Gambia Science Students Association for inviting me to give a talk. University of Edinburgh hosts Pathogens and Global Health PhD program for funding my PhD. And most importantly, I'd like to thank Drosophila melanogaster for being such a fantastic model organism. So on that note, let us keep calm and ask me questions.
Thank you very much, Oumie, for that fascinating talk. I've learned so much about Drosophila. Can we all give her a round of applause before we go to questions? That was amazing. Your research is also interesting. And you know, lucky for you, you can go to Gambia and eat mangoes from time to time. I really miss the mangoes. So before we go to questions, I just have a few. If you have any questions, please put them on the chat so that Mustafa can post them on the floor. And after questions, we'll have some closing remarks by the SG of the UTG Science Student Association. Thank you again, Oumie, and we're going to take questions.
Once again, thank you very much, Oumie, for that wonderful talk. So the first question is from Lamin Drame. He asked a very good question. He said, what is the lifespan of a virus outside its host?
Oh, that's a good question. I think that varies from virus to virus, actually. So I'll just say it varies.
The other question is from Idris Ajay. So he says, in measuring virulence, did London, I think the person that you reference, account for different virulent responses? And also, how did they measure virulence?
Good question. Let's go back to the paper where I have the slides. So Ben Longdon, yeah. So he measured virulence following mortality. So he just looked at the number of flies that died after infection. So he did it over 16 days and was following the flies and each time measuring how many of them died. So that's how he measured virulence.
Okay. And another question from Dam Khan. What could be the reason of the disparity in response to viral infection based on sex?
So obviously there are sex differences in generally. Males and females have different gene expressions. And secondly, because of sometimes when they're infected with viruses, females are more able to, they handle it better. It's just been found as sex, like females are generally in some viruses respond better, able to handle the infection more than males. So sometimes you see actually males having a higher immune response as a result of virus infection. And because females lay eggs, males don't, sometimes when females are infected with viruses like the Calophaea virus, the females actually lay less eggs and are putting more energy into survival and less into reproduction. So it just happened that males and females are different, have different gene expressions and respond to viruses differently. They even respond to aging, all of those is just different between the two sexes. I hope that answers your question. Or did I not get the question?
So from Shirfo Cisse, so this one is quite interesting. So it's like, can you delve more into climate change being a factor for viruses jumping from one species to another? And he also spoke about viruses being biological weapons and whether that's a conspiracy.
I know this one conspiracy. I think I can't remember, but it was the 2009, I can't remember correctly. I think it was the 2009 like H1N1, and some were saying it was like put out by Russia. And so there are conspiracy between scientists that maybe Russia put it out. But the coronavirus was not manufactured in the lab. Andrew Rambo did work to show that that did not happen. And what was the other question that he asked before that?
Yeah, I said how does climate change impact the shift of viruses from one host to another?
So basically, because as climate change, sometimes you might have more vectors and the number of vectors might change, and vectors can enable host switching. So that's how climate change, one way that climate change can lead to emergent infectious diseases.
And this is from Omar Jangha. Was virulence in any way related to viral load?
Yes. Evolutionary theory actually couples virulence to viral load. Although there are some caveats to that theory in the sense that it's just not viral load. Sometimes it could be the host response can also affect virulence. But yes, virulence from an evolutionary perspective is tied to viral load.
So there's this one from Schaefer again. He said HIV has been around for so long. So what is the challenge that the scientific community is facing in developing a vaccine against it? And it says, remember, I'm not a health scientist. So I think you
okay.
you could break it down.
I'll try and answer that because I'm more of an evolutionary biologist now. My biochemistry days and molecular biology days are long gone, but HIV mutates so quickly, the RNA virus, that it's almost impossible to make a drug for it. But the PrEP, like the medication is amazing because people with HIV actually live longer now than people without HIV. There's the antiretroviral, the PrEP, and all of those. So yeah. Was that the question? Can you read the question again? I think I did answer that.
Yeah, I'd just like to add.
Yeah, it says that what's
I'd just like to add, for even us health scientists find this very, very challenging and contentious. We're still struggling to really understand why none of the vaccines being tried aren't providing long-term efficacy. Also, there's the issue of safety, right? So there's so many different reasons why vaccines might not work.
Okay, thank you. Yeah.
And also to add on that, we're still trying to fully understand the immune response to viruses. So even if people die from HIV, once they do a post-mortem study, you still see that they still contain the virus. And it kind of shows that the virus still resides in a lot of tissue. So even if you find a therapy like antiretroviral treatment, it clears the virus in blood, but you still have a lot of viral load in tissues. And it's kind of a challenge to get rid of this as well. So it's kind of really tricky for scientists in general.
Okay, thank you.
Next question is from Idris Ajay. He says, this might be ignorant, but just curious if viral load is equals to density of virus in the host.
Yeah, it's just the number of viruses. How much is that?
And no question is ignorant or stupid, by the way.
Exactly.
We're all learning.
Yeah.
Yeah.
So it was found by
qPCR.
It was just a method that he did to measure viral load.
Yeah, and this is for KM Wada. I'm not sure if it's Kathy. What do you think is the most useful bioinformatic tool to compare the genetic data you work with, and why?
Ooh, that's so basically, it's so long, but I shortened it. But I don't know like what, because let me see if I can you see my screen?
Yeah.
Okay, so basically, when I say I get, like, this is simplified, but each of these ones has a bioinformatic tool. Like the separating the viruses, I need to map it. I use STAR because it can handle alternative splicing. And there's also backward switching that I came up with the mathematics that I need to work with. And I do some de novo build using Trinity. There's so many tools. And I use Dream to, so it depends on what stage she's asking. Because these figure, I got it using TopGo, and this was done with Dream, and yeah, it depends on, because literally each square that I have here has about 10 little squares. So the maths and stats took me months of learning starting. So yeah, it depends on what stage that Kati is asking.
And this is from Mamadou B. Jala. Are the challenges faced in HIV related to herpes virus, which is very rampant?
I don't know, actually. There's someone working HIV and herpes that can help me. But I do know HIV does affect your immune system, so I guess it makes you more susceptible to viruses. But
well, actually, there's a lot of relation of HIV. Sorry, go ahead. Go ahead, Nana.
No, go ahead. I'll go after you.
Yeah, there's a lot of comorbidities associated with HIV, such as like opportunistic infection. So opportunistic infections are infections that wouldn't normally occur if you were immunocompetent. So you have a lot of fungal infection and other diseases such as TB. So you become more susceptible to other infections as a result of HIV. So as a result, you could also have other infections, which would include viruses. But whether specific to herpes, I'm not sure. I've not seen anything with regard to that.
So to add to what Mustafa said, I've worked on herpes viruses, and it's a huge family. There's at least 6 different types of herpes simplex, and there are interactions between different viruses if you happen to be infected with more than one. And we find that HIV, obviously, because it attacks the immune system, it makes you more susceptible to any other disease or infection, and your prognosis is a lot worse if you have HIV with something else. So there's a lot of interactions comorbidity poses, and I think scientists are still trying to figure out exactly what happens, but the most studied ones are HIV with TB, because those tend to have worse outcomes.
Seems like that was the question. So are there any more questions, please? Okay, yeah. This is from Abdullah Ngom. Any idea if there is or will be a vaccine for HIV, because of there is PrEP and PEP? Okay, I don't know what he meant by that, but it's like, is any idea if there is or will be a vaccine for HIV? So maybe if you could shed some light on any works that's been done.
I don't know anyone working on PrEP. They've been working on trying to get a vaccine, but I think with the PrEP, it's worked really well, and it has saved a lot of lives in the UK. Especially, I was reading a paper for men who have sex with men and also with women. Like, it's literally saved lives. So the PrEP works. So I don't know. Maybe someone else works on vaccines can tell us, because it's very difficult to find something against HIV, because it's an RNA virus and it mutates so quickly, which is why evolutionary biologists, there'll be a PhD student joining our lab soon to actually say, is there even a host virus coevolution? Because if you really think about it, viruses evolve at a faster rate than the host. So this whole Red Queen hypothesis that evolutionary biologists have been going on for 60, 70 years, is it actually happening? Because, yeah, it evolves so quickly. Maybe someone else works on vaccines that can help us. I don't know.
And also, one of the reasons why it's been really hard to make a virus against HIV is because the glycoprotein, which is very important, is actually being covered. And it's only expressed during the latter stages of when it's infecting an actual immune cell. So even if you mount a response, it tends to expose it to antigens, which are not really necessary for the survival of the virus. So technically, you just mount an immune response, but you wouldn't actually get rid of the virus. So that's what has made HIV a pain so far.
Okay, are those all the questions? Are those all the questions?
Is it safe to
Anyone? Sorry. I
so,
think from the chat, unless there's anyone. So please keep the questions coming in.
Yeah, so if Oumie doesn't answer them now, we can always share the answers later. But to close off,
Exactly.
I have one final question for Oumie. And so, besides enjoying the mangoes and all of that, what has been, your favorite aspect of doing a PhD? And do you have any words of advice to go with that for those young Gambians like yourself who are in university at the moment and hoping to pursue whether it's master's degrees or PhDs, just something very, very, very brief based on your own experience?
So the first question is, what have I enjoyed? I think I like the fact that I'm being paid to just learn. I just love that. Every single day I wake up, I set my own time, and all I have to do is just learn. And I've really enjoyed learning how to code and being able to do bioinformatics. So yeah, research. I enjoy learning, and then also seminars, meeting different scientists, reading and discussing, and broadening my horizon. And an advice I'll give is always seek mentor. To me, that has been the best, just speaking to different scientists, especially Black women. Like sometimes when I have questions, I do ask Nene and there's Furaha, there are PIs. By having female mentors, especially Black women mentors, has really helped my career. And another thing I'll say is just, people keep saying work hard, Work smart. Isn't that just the normal? But just keep pushing, I guess. That's the advice I'll give. Keep at it.
Thank you very much, Oumie, and for answering the question so brilliantly. And yeah, we're really.
I think there's one more question. I don't know. Do we have time for one more question? No.
I think that's more on that's more vaccine side. Can we answer those later? Where I'm trying to stick to time because 1 o'clock. But yeah, we're really grateful that you joined us today, and I'm sure a lot of the students out there would want to hear more from you, and so we'll keep the dialogue going. Before we close, we have some closing remarks from the UTG Science Student Association Secretary General. I think Mr. Sekou Kuyateh, is that right?
Yeah, Kuyateh.
Sekou?
Yeah, Kuyateh.
Kuyateh, right, exactly. So Sekou, I'll hand over to you before we close. Thank you.
Thank you very much, Dr. Nene Salah, for that brief introduction. I would like to thank the Health Science for Gambia for this great initiative. First of all, my name is Sekou Kuyateh, like you rightly mentioned. I hope I am visible.
Yep.
All right, thank you. Okay, on behalf of the Science Students Association, I would like to thank the Health Science for Gambia for organizing this, and special thanks to Ms Oumie Kuyateh for that brilliant and educative session with us. I believe most of the students have learned a lot, and what is gained here today will be extended to the society so that many can benefit, especially those that are not in attendance here. And I believe students here will be willing to have more of this session with Health Science for Gambia, and I believe you are always ready to serve our community, especially the university students, because it is said that when knowledge is redirected to solve societal problems, then it is qualified to be called knowledge, and that is exactly what you people are doing. So we are very, very grateful, and we thank you always for coming out to share what you are learning, and we believe what we are also doing here will go back to the society and extend so that we can all benefit. So on that note, I will thank you. And before I conclude, I would like all of us to unmute our mic and say together, we can do it. I would really appreciate that on behalf of the Science Students Association. Can we do that?
On three, maybe?
After the count of three? After the count of three. 1, 2, 3. We can do it. Thank you so much. I like that.
Thank you, Sekou, for that. So please stay tuned for our next seminar, which is in March, and it's going to be given by one of our colleagues, a brilliant scientist. I don't want to spoil the surprise by telling you who he is now, but he will be talking to us a lot about the COVID pandemic and epidemics and pandemics in general. And all I can say is, you know, he's also a son of the Gambia and is currently working at Harvard University now. And yeah, it's going to be brilliant. So stay tuned and
I don't know. But anyway, it's fine.
Since you already spilled the bean.
Sorry?
I say it's like you already spilled the beans.
Exactly.
Yeah, but it's going to be fantastic.
Exciting.
Yeah, exciting.
All
All right.
right.
Have a lovely
I think people will need Google.
Bye.